Buckle up and let's go on a whirlwind road trip through the heart of central metabolism. We'll start with taking the main road, straight shot from glucose through glycolysis, through the citric acid cycle, and the electron transport chain with those electrons generating ATP through ATP synthase. But then we'll also take some detours, we'll go some side routes, and we'll think about how cells decide which of these routes to take. Are you ready? Let's dive in. First things first, let's learn the rules of the road. Each of these represents an intermediate, a metabolite that is being converted between. We call them intermediates because they're intermediates in a variety of different pathways. You can see that rather than straight pathways, we actually have more of an interconnected net. There are key things, though, that if you know, you can figure out a lot about the map. If we have two arrows next to one another going opposite directions, that's indicating that something is reversible. A reaction is reversible. And so this is like a two-way road that you can go back and forth on really easy. How do you know which way to go? Well, that's just going to depend on the concentrations of the intermediates on either side. So if you see one of these double arrows, these two-way arrows, easily go back and forth. Great. But then you have some one-way roads. You have some places where you have single arrows. So they're either by themselves or they're next to one another but separated, and each of them has a different enzyme that's catalyzing it. So in purple, these are the names of enzymes. And the enzymes are able to catalyze these reactions, basically make them happen. Or if you inhibit the enzyme, then they won't happen. So it's more like they are able to remove a stop sign, but they can't control the fact that it's a one-way road. So enzymes cannot change the direction of a reaction, which is why if we want to go back from one of these one-way arrows, we need to use a different enzyme that catalyzes a different reaction that has the overall effect of reversing the actions of this enzyme. So these are going to serve as sort of go, no-go steps. These steps where you have these single arrows. And so you're going to see that we're going to regulate those steps really, really highly. Because if we want to go back from those steps, well, then we're going to have to take a detour. And sometimes that detour is actually going to be really costly. And it might even involve multiple steps. So we're going to regulate these steps depending on what we have and what we need. And so if you see something in green, that's going to be saying it's activating the enzyme. And if we're activating the enzyme, you think green light. If we're inhibiting the enzyme, we see things in red with this X. That's saying something is inhibiting the enzyme. And so now we have a red light. But remember, that was a one-way street. So red light, green light is still only going to be one way. And so we want to regulate those very highly. Anywhere else, you can kind of just go back and forth from fairly easily. So let's start on the main route. We have the glycolysis. Glycolysis is going to represent the breakdown of glucose to give you pyruvate. And so the glycolysis is this green direction. Gluconeogenesis, if we want to go the reverse, we have to take detours along those irreversible steps. But for now, we're just going to start with glycolysis. Glycolysis is going to be a series of rearrangements and oxidation reactions. In the oxidation reactions, you're going to be passing electrons from these organic molecules that you're breaking down onto NAD+ to give you NADH. The electrons later on, we'll see that those electrons are actually going to be used in the electron transport chain. A key thing to remember is oil rig. Oxidation is loss of electrons, reduction is gain of electrons. And another key thing is that passing electrons from something that wants it less to something that wants it more is going to be energetically favorable. It'll produce energy. And you can use the energy that's produced in order to do things. In this case, the electron transport chain is going to pump protons from an inner compartment to an inner membrane space, create a gradient of protons that's then used to power the synthesis of ATP. So you pass electrons off through these molecules as you break them down, You take those electrons through the electron transport chain, pump out protons to build a gradient, use the gradient to power the production of ATP. So ultimately, we're going to be able to take this NADH and use it to make ATP. But first, we got to break down the glucose. And well, first, we've got to keep the glucose inside the cell. The glucose is going to enter the cell through a glucose transporter. Different cell types have different glucose transporters with different properties so that you can kind of give more or less glucose to various cells. But that's just one level of regulation. When the glucose enters the cell, well, now if you decide you want the glucose, you're going to trap it. You got to trap it by phosphorylation. So hexokinase is a version called glucokinase in your liver and pancreas. This is going to trap the glucose inside the cells by phosphorylating it so it can't sneak out. It really, really traps it in the case of cells other than your liver and your kidneys, because most cells do not have the enzyme that's needed in order to remove the phosphate group. That would be glucose 6-phosphatase. Most of your tissues don't have this, but your liver and your kidneys do. This is really important because your liver and your kidneys are therefore basically the only tissues that can release the glucose from the cells. So the other cells, they have to decide, okay, do I trap the glucose in the cell? And they can trap the glucose in the cell by phosphorylating it. But you don't have them trapped too much, so the glucose 6-phosphate is actually going to inhibit the hexokinase enzyme, except for the version in your liver and your pancreas. So that's going to allow your liver to kind of serve as the glucose sensor and to keep bringing in the glucose. And it'll allow your pancreas to keep an eye on sugar levels so it knows whether or not it should be shipping out insulin or glucagon or what to do. Because those are going to help regulate which of these pathways you're going to go down. So we have the glucose, we phosphorylated it, we have glucose 6-phosphate. Now you can see that there's still a lot of options that you can have. We're going to continue down the central route, follow these green arrows directly down. However, we could also alternatively go down the pentose phosphate pathway, and that is going to allow us to do things like make DNA and RNA. We can get NADPH, combat oxidative stress, build up lipids. We'll come back to it in a minute, and we're actually going to encounter multiple places along the path to glycolysis that we can enter into the pentose phosphate pathway if we want the RNA. But if we want the NADPH, we got to choose up here. If you're in your liver or your muscles, another fate is that you can basically hook up those sugar units to form a storage form of glucose called glycogen. We call this process glycogenesis. This happens in your muscle and your liver. And then what happens is that when you want glucose or you want the energy from glucose, you're going to break that glycogen down through glycogenolysis. When you break it down, you get glucose 1-phosphate, which you can convert to glucose 6-phosphate. Your liver, as we said, can remove the phosphate group in order to get glucose that can get released. Your muscles don't have this enzyme, glucose 6-phosphatase, so your muscles are going to be stuck with the glucose 6-phosphate, but now they can go take that through glycolysis in order to get energy. So the muscles and the liver both store glycogen, but the muscles store it to use the energy for themselves, whereas the liver stores it in order to be able to make glucose rapidly to let it out. The liver can also do gluconeogenesis and make it from non-sugar parts, but that's going to take longer because we got to go this whole pathway, as opposed to just kind of having it pre-stored, chop it off, and let it out. Your kidney can also let out glucose, but your kidney doesn't store glycogen, so your kidney's got to make it from scratch. So your liver is going to really pull the brunt of the work when it comes to regulating your blood sugar, and it's going to be influenced by insulin and glucagon. We got a lot of blood sugar, then insulin says, let the glucose in. Not a lot. Glucose is gone. Your pancreas releases glucagon, and that glucagon is going to inhibit you doing things like storing the glucose as glycogen, and instead activate you releasing the glucose to go get your blood sugar up. And that's just one place we see insulin and glucagon acting. For now, we continue down. We just shift things around and we get fructose 6-phosphate. Again, we have multiple options coming from our fructose 6-phosphate. Now we can get back into the pentose phosphate pathway. We can go and we can make amino sugars through the hexosamine pathway. We can go and make this regulatory molecule fructose 2,6-bisphosphate, which is actually going to regulate this protein. But for now, we're just going to continue forward. We're going to continue forward using this phosphofructokinase 1. And note that once we continue forward, well, we have a lot fewer options. We can't go back to all of these pathways easily. There's a separate enzyme that'll allow us to go back from this, but in a different way, just chopping off that phosphate group. And so this step is going to be a one-way road that we want to really, really highly regulate. And it's a one-way road that we call the committed step of glycolysis. If you're at fructose 6-phosphate, you can still go back and forth to glucose 6-phosphate, so you can get into glycogen. You could get into the top of the pentose phosphate pathway. From fructose 6-phosphate, you can get to the hexosamine pathway. You can get these other routes. But once you go and you commit yourself to that PFK, well, now there's fewer options. So now you've got 2 phosphate groups on there. And so now you can split it in 2. And when you split it in 2, each of those pieces is still going to have a phosphate group. And each of those pieces you can convert between. You can do so readily using this isomerase. So when you have molecules that are similar in structure, they just kind of using to shift things around a bit, those isomerization reactions, those are typically going to be easily reversible. And so triose phosphate isomerase can interconvert between these 2. So we split the molecule in 2 with aldolase. We started with the 6 carbon. We split it into 2 3 carbon pieces. And each of those pieces has a phosphate group, and you can interconvert between these pieces. But only one of these pieces is actually going to be able to continue on through glycolysis. That's going to be this glyceraldehyde 3-phosphate. And so what's going to happen is you're going to use the glyceraldehyde 3-phosphate and pull things this direction. So the dihydroxyacetone phosphate will then get converted to the glyceraldehyde 3-phosphate and taken through glycolysis, if this is the pathway we're taking. I say if this is the pathway we're taking because there are also other routes that you can go. And so if we wanted to go back from this, we would pull things in this direction. Glyceraldehyde 3-phosphate dehydrogenase is going to now do our first oxidation reaction, the first step of our energy payout phase of glycolysis. In this first part, we actually had to invest energy. We had to spend 2 ATP by the time we got here. But now we're going to see that we're actually going to make ATP, but first we're going to make NADH. And we're going to do each of the things down here twice, because we're going to do it once with the glyceraldehyde 3-phosphate and then once with the glyceraldehyde 3-phosphate that we got from converting the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So everything down here is going to happen twice. And so we're going to generate an ATP here and an ATP here, but then that's going to be equal to do it again, so you can do ATP and ATP, so you get 4 ATP minus the 2 ATP, so you get 2 ATP total, and then you're going to get an NADH, and that NADH is actually then going to be more useful for you in terms of the bang for your buck at ATP. So we'll cash that in later. For now, we just continue forward, and all of these steps are going to be reversible until we get to this pyruvate kinase step. And so now we have this pyruvate kinase step where we have a one-way road, and you're going to have to take a detour to go around it. And that detour is actually 2 blocks. You've got to go through 2 different enzymatic steps to go backwards, and you're going to spend an ATP and then spend a GTP in order to do so. And you actually have to use a separate intermediate as well. And so this step is going to be very, very energetically favorable in the glycolysis direction. It's going to be the big bang at the end of our pathway. So we got to really spend some energy to go back from it. And so we're going to really want to regulate it as well. And so F16BP will actually see that this molecule all the way up here, if you've got a lot of this coming in, it's going to activate this pyruvate kinase so that you can help drive this whole pathway forward, because this is going to be irreversible, but these are not irreversible. And so by kind of regulating this, you're pulling things forward. But if you have too much energy, well, now you're going to inhibit this kinase. So ATP, if you've made enough ATP, NADH, you've got a bunch of things, then what's going to happen is that you're going to say, okay, we've had enough, and you stop pulling down glycolysis and maybe go some of these other routes. And we'll get to those other routes in a minute. But right now, we want to think about, we're left with pyruvate. Now, pyruvate, it still has a lot of oxidation potential, and we're going to oxidize it with pyruvate dehydrogenase. Pyruvate dehydrogenase, this step is now going to give us an NADH that will cash in and leave us with acetyl-CoA. Note that this is happening now in the mitochondria. So all of this glycolysis was happening in the cytoplasm, but now we're in our mitochondria. And I emphasize this for a couple reasons. One is that the citric acid cycle happens in the mitochondria, and the other is that electron transport chain happens in the mitochondria. And the other is that acetyl-CoA cannot just enter and exit the mitochondria. And so you'll have separate pools of acetyl-CoA in your mitochondria that are used for the citric acid cycle. And then you'll have pools of acetyl-CoA in your cytoplasm that can be used in order to make lipids. And the NADH also cannot enter or leave the mitochondria. And so we actually have to shuttle the NADH in that we made from glycolysis. But right now we're in our mitochondria with our acetyl-CoA. We're going to take this acetyl-CoA and enter the TCA. So the TCA, the tricarboxylic acid cycle, the citric acid cycle, the Krebs cycle, whatever you want to call it, it's the central hub of metabolism. You can see it interconnects with a lot of different pathways. The TCA, here we've got a roundabout. And so we can go off at different exits. We can do different conversions, various things like this. It interconnects with a lot of different pathways. You're converting these molecules between one another, and each of those molecules as intermediates can also then have different fates. Many of the steps are going to be easily reversible, whereas others are going to be irreversible. And those irreversible steps are the ones that you're going to want to regulate more for the reasons we discussed before. Right now, we're going to use the TCA in its full entirety, going all the way around, in order to get the most oxidation out of our molecule, generate NADH and NADH and FADH2. What we're doing is when we're doing these oxidation reactions, we're passing the electrons onto those carriers that want them more, and then those carriers are going to take them to molecules that want them even more. So right now, this is all happening in our mitochondrial matrix. And then outside of the matrix is going to be our inner membrane space. And then there's going to be another mitochondrial membrane and then the cytoplasm. In order to get ATP made, we're going to have our NADH come through this complex 1. It's going to pass these electrons off through complex 1 to a molecule called Q. And this is going to be energetically favorable, and so favorable you can pump protons out of the mitochondrial matrix and into the inner membrane space. Then Q is going to take them to complex 3, and it'll pass it through complex 3 to cytochrome C, again pumping protons. And then cytochrome C will take it to complex 4, again you're pumping protons. You're pumping them up so that you're have a lot of protons out here. And these protons out here, the only path through is through this ATP synthase protein. And when they flow through this ATP synthase protein, that produces energy that is used to put a phosphate group onto ADP to give you ATP. So you get ATP from the passing of those electrons. You also generated an FADH2 in the citric acid cycle, and that was actually happening directly at this complex 2. Complex 2 passes the electrons onto Q, but it doesn't pump anything. So you get more from your NADH than you get from your FADH2. As long as that NADH was generated in the mitochondria. When you're generating NADH in your cytosol, then you can't take the NADH in. You can take the electrons from it in, but NADH can't get through the membrane. So what you have to do is you have a couple options. One is the malate aspartate shuttle, and you sneak the electrons in that way. The other is the glycerol 3-phosphate shuttle, where you sneak the electrons in this way. Not going to go into the details, but basically you put those electrons onto another molecule that can get in, and then use that other molecule, then you reverse things on the other side. In the case of our glycerol 3-phosphate shuttle, here you're actually going to be passing to FAD, which passes it to Q. And so that case, you don't have enough energy to pump. But with the malate aspartate shuttle, you are able to get that full yield, go in at complex 1 with your NADH. And so that's how you generate ATP from this pathway. But sometimes you have too much energy. And so if you're breaking down a bunch of glucose and now you're building up a bunch of, we want to say, okay, let's stop this process. What else can we do? So if we look at kind of our road signs, we'll see that a lot of these enzymes involved in these catalytic reactions are going to be inhibited by ATP. And so we saw that pyruvate kinase was inhibited by ATP, as well as pyruvate dehydrogenases, citrate synthases. We have isocitrate dehydrogenase, and is basically these enzymes that are controlling those one-way roads that are driving you through this catabolic pathways are going to be inhibited when you have a high buildup of the ATP. But if you need to make more ATP, well, now we have enzymes that are going to be activated by ADP, enzymes that are going to be activated by AMP. And that AMP is a signal of the low energy status. You also see that phosphofructokinase 1 was activated by AMP and ADP. So if you need ATP, you want to break down the glucose. But if you don't need more ATP, well, now you want to shut things off. And when you shut things off, now things are going to go through those reversible pathways to other fates. So let's look at some of those other fates. Let's go back and think about kind of how could we make glucose? The pathway of making glucose from pyruvate is called gluconeogenesis. So we have these 3 detour points. We have this one, we have this one, and we have this one. If we want to make glucose, but we had one of these things, we just go this way. But what if we want to take parts from the mitochondria and get them to make glucose? Here we have another instance where one of the molecules that we need can't leave the mitochondria or can't go through the mitochondrial membrane. We see that we have oxaloacetate as an intermediate in gluconeogenesis. And we also have oxaloacetate as an intermediate in the citric acid cycle. But the citric acid cycle was taking place in your mitochondria, and this part of gluconeogenesis is taking place in your cytoplasm. We can't take oxaloacetate out, but we can take malate out. And malate can be easily converted back to oxaloacetate. So we can take malate out of the mitochondria and put it into the cytoplasm and then convert it back to oxaloacetate and then use that for gluconeogenesis. And so that is how we could get pieces from the citric acid cycle into the gluconeogenesis pathway. And then you could go up the gluconeogenesis pathway, but a lot of tissues are just going to go up to the glucose 6-phosphate, and then they're going to get stuck because they can't release the phosphate, so they can't let out the glucose. The glucose 6-phosphate, now let's look and see what we can do with it. If you're in your muscles or in your liver, you can go and store it as glycogen. And so in order to store it as glycogen, we just do an isomerization, shift things around, and then we're going to use UTP here, and then we're going to make UTP glucose and then make glycogen. Note that this is going to be stimulated by insulin. So insulin is released in response to high blood glucose, and it's going to be saying store that. And it's going to be inhibited by glucagon. And glucagon is saying there's no glucose in our blood, so let's make some glucose. But let's use the glucose. So we don't want to store the glucose, we want to use it. And so that's why glucagon is going to inhibit this making and stimulate the breakdown, and vice versa for insulin. And they reciprocally regulate one another. You could also, if you decide you want to go to the pentose phosphate pathway, You can use this in order to generate NADPH. NADPH is a reducing agent, and it's similar to NADH, but we keep kind of separate pools of it in NADH, and that way we can keep the NAD plus ratio where we have a lot of NAD plus to use for oxidizing reactions and a lot of NADPH to use for reducing reactions. And the reducing reactions that we use NADPH for include the making of fatty acids. And this NADPH is also important for combating reactive oxygen stress. And so if you need NADPH, you want to enter the pentose phosphate pathway in this oxidative phase. But however, you can then enter the rest of it, the non-oxidative phase, from multiple points. From fructose 6-phosphate, you can go enter that way, or glyceraldehyde 3-phosphate, you can go back and enter the pentose phosphate pathway. And you can shift things around because you've got these easy two-way streets, but you can't go back from the oxidative phase. You can't go back from this direction. And so instead, you just kind of move around, shift things around. There's a lot of isomerization and kind of transferring carbons from one molecule to another, but not energetically costly. And so you can go back and forth between these. And so this non-oxidative phase of the pentose phosphate pathway is going to be useful for doing things like getting you nucleotides and DNA and RNA. And so if you want to replicate your cell, say, you're going to need lipids, so you need the NADPH, but you're also going to need to replicate your DNA, so you're going to need the non-oxidative phase. But depending on how much you need one versus the other and things like this, you can use some of it, get the NADPH, and then go back into glycolysis to get energy, various things like that. So cells can do things very strategically. And whether or not they go down this pentose phosphate pathway's oxidative phase is going to be determined by whether or not you need the NADPH largely. And so if you've already got a lot of it, then you want to inhibit this glucose 6-phosphate dehydrogenase. But if you don't, then you want to activate it. Now you decided that you're going to shift things around to fructose 6-phosphate, and here you're just kind of controlling the concentrations and stuff. Now we have this kind of option where we could go this hexosamine pathway. And this hexosamine pathway is going to give you those amino sugars that you can use to make glycoproteins and glycolipids. So stick these fancy sugars that have these amine groups onto other molecules. That's how you could kind of get to there. Fructose 6-phosphate, if you've got a lot of it too, what you can do is you can use phosphofructokinase 2. And that is actually going to then take this fructose 6-phosphate and put a second phosphate group on it to give you this regulatory molecule, fructose 2,6-bisphosphate. And that 2,6-bisphosphate is then going to serve as a negative feedback on this phosphofructokinase 1 enzyme. And so if you have a lot of this fructose 6-phosphate, some of it is going to be used by this phosphofructokinase 2 to generate this regulatory molecule that's going to stimulate your pathway in this direction. And you can see that this is going to be stimulated by insulin. So insulin says there's high blood glucose, let glucose in and use it. Glucagon says glucose is gone, better make some. And so you have that this is going to be stimulated when you've got a lot of glucose. You want to go forward this pathway. And this step is going to help you know that you've got a lot of glucose to go forward this pathway. It's going to help this enzyme know. Also, this phosphofructokinase 2 is activated by X5P, which is going to be one of the things in the pentose phosphate pathway. So if the pentose phosphate pathway is going strong, then maybe you want to put some of it into glycolysis too. And so here you're going to make more of the regulatory molecule that's going to activate this molecule to drive things this way. So you've got a lot of hormonal regulation going on up here to help dictate which way the glucose products are going to go. Now we think about we're farther down. We want to think about now we've got these smaller pieces, and we can interconvert these pieces, and we can take them from different pathways that all use those same pieces, like Legos from different kits. We see that here we can actually feed in addition to being able to kind of take these 3 carbon pieces in from the pentose phosphate pathway, we can take in 3 carbon pieces from glycerol. And so glycerol makes the backbone of triacylglycerols, so storage lipids. You hook them up with this glycerol backbone, as well as some phospholipids. Basically, you stick these fatty acids onto a molecule of glycerol. When you break down those fatty acids, you get acetyl-CoA, which you can then go through the citric acid cycle. Or if you have a lot of it, you can make ketone bodies that the liver can ship out to give energy from fats to tissues like your brain that really need energy when you don't have enough glucose. But that leaves you with the glycerol backbone. The glycerol backbone, you can then use glycerol kinase, phosphorylate it, and then you're going to basically take it and convert it into dihydroxyacetone phosphate. And that is then going to allow you to take those carbons and their electrons in through the mitochondria where they can be broken down further. This also glycerol 3-phosphate dehydrogenase. This is part of this enzyme. There's a cytoplasmic and a mitochondrial version of it. And those are involved in the glycerol 3-phosphate shuttle, where we can actually get NADH that was generated into the cytoplasm. We can sneak the electrons from it into the mitochondria. But for now, we just think about, okay, now we've come in. Where else can we enter the pathway? We saw how if we wanted to make glucose from the citric acid cycle, we'd actually take out malate, and then we'd use that in order to go out of the cytoplasm, convert that back to oxaloacetate, and make our glucose that way. And now we have that pyruvate. Pyruvate, here we can actually convert pyruvate to alanine, and we can convert pyruvate to oxaloacetate. This conversion to oxaloacetate is really important because if we remove things, like if we remove malate, well, now we don't have any malate to turn into oxaloacetate to make combined with acetyl-CoA in order to make citrate. And so this is what we call an anaplerotic reaction, where we're generating a pathway intermediate when we remove something. And this lets us remove things and still have the cycle be able to keep going because we're adding it in from other sources. So as long as we keep adding carbons into this pathway, we can then take out other molecules from the pathway. And because they're all going to convert as a cycle, if you take one thing away, but no matter where you add back in, you're still going to be able to make the cycle keep running. You can feed in from not just the pyruvate, but also from amino acids. So glutamate will give you alpha-ketoglutarate. Aspartate will give you oxaloacetate. Then you can also take aspartate and use it to help you remove amino groups as urea. Odd-chain fatty acids, when you break those down, you're left with the extra 3-carbon piece. Propionyl-CoA, which can be converted to succinyl-CoA, which can then enter the TCA that way. And so lots of ways into and out of the citric acid cycle. And that's really important because that is going to feed into a variety of different pathways depending on your needs. Another molecule that's useful is citrate. Say you want to make fatty acids. Making fatty acids happens in the cytoplasm. When you get acetyl-CoA from the pyruvate dehydrogenase, that's in your mitochondria. And the acetyl-CoA can't leave the mitochondria. So what we got to do is we've got to actually convert that acetyl-CoA to citrate by joining it with oxaloacetate. And then we'll ship out that citrate, cut off that oxaloacetate with citrate lyase. And now we've got our acetyl-CoA in the cytoplasm. And there we can use it to make fatty acids. We can use it to make cholesterol and steroids, all those things that happen in the cytoplasm. So we've snuck out the equivalent of the acetyl-CoA. Just like we can sneak in and out malate to get oxaloacetate, and we could sneak in and out electrons through the malate aspartate shuttle and the glycerol 3-phosphate shuttle. So these sneaky paths that'll allow us to take detours, to go around bottlenecks and things like this. And so those are the basic processes of central metabolism. You've got your glycolysis, gives you pyruvate from glucose. You take that through the citric acid cycle. You're passing off electrons. You're passing those electrons through the electron transport chain, pumping out protons, building a gradient, making ATP. But that is just the main thoroughfare. You can also go and you can store the glucose, glycogen. You could go and you could take it down the pentose phosphate pathway to get NADPH, to combat oxidative stress, and to do things like make lipids. You could use the pentose phosphate pathway's non-oxidative phase in order to make pieces for your DNA and your RNA and your coenzymes. You can use pieces from the glucose in order to make modified sugars through the hexosamine pathway, regulatory molecules. You can feed into the pathway from your lipids so you can go further down and make more energy. You can feed into the pathway from malate that you got from the citric acid cycle. And from the citric acid cycle, you could go and you could take pieces out to do things like make lipids. You take out your citrate, you can go make some lipids. You take out alpha-ketoglutarate, you can make glutamate. You take out oxaloacetate, you can make aspartate. All these various things that you could do with these pieces. And as you do so, as long as you're going in the oxidative direction, you're going to be generating NAD+. And if you're going through these oxidative reactions, you're going to be generating NADH, which you can then use in the electron transport chain. The electron transport chain, however, it's going to require that you have a mitochondria because this is happening in the mitochondria, and some cells, like red blood cells, don't have this. And it's going to require that you have oxygen. And it's going to require that you've got ADP and NADH. If you don't have those things, if you don't have oxygen, say, now what are you going to do? What you're going to do is you're going to go through anaerobic fermentation. So if we look at pyruvate dehydrogenase, it's inhibited by NADH. And NADH is going to build up if the electron transport chain can't go because there's no way for it to go. And if we look at glycolysis, we can see that glycolysis is actually going to require NAD+. And so it's going to require NAD+, but if we can't take the NADH and pass the electrons off further, then we're going to build up the NADH. We're going to run out of our NAD+, and we're not going to be able to do glycolysis. And we're actually going to inhibit the glycolysis by inhibiting the pyruvate kinase. So what are we going to do? What are we going to do is we're going to find another place we can pass those electrons to. You can pass the electrons from NADH onto pyruvate. That'll give you lactate. And it's done by lactate dehydrogenase. So that's anaerobic glycolysis. We go through glycolysis, and then we pass those electrons off from NADH onto pyruvate to give us lactate. And now we can go, we've got our NAD+ again. We can keep this going. We can keep glycolysis going and still be getting the energy from the ATPs, even though we don't get the energy from the NADH. And now we can take this lactate and we can ship it out to our muscles, and our muscles will convert it into pyruvate, and they'll get the NADH that they then can use to do things like make you glucose and ship that glucose out to you, because your liver can actually remove that phosphate group and let out glucose. Your liver is going to store glycogen, and it's going to be able to make glucose, go the full gluconeogenesis and let it out. Your kidneys can do gluconeogenesis and let out the glucose, but they don't store glycogen. So your liver is really the key thing, the key organ for helping control the blood sugar levels. And there you go. Hope you enjoyed your trip.